Most proteins don't work alone. They pair up, they form complexes, they snap together into molecular machines with moving parts, active sites, and feedback loops. And until today, the largest computational biology database on Earth, the one that predicted the shape of 200 million individual proteins, couldn't really show you any of that. It could show you the pieces, but not the assembly. But today, a consortium spanning Seoul, Cambridge, Mountain View, and Santa Clara added 1.7 million protein complex predictions to the AlphaFold database. So for the first time across 20 species, humans, mice, yeast, tuberculosis bacteria, the database shows how proteins actually function together. And that shift from isolated parts to working machines sits at the center of a day where distance between observing biology and designing it compressed in three separate directions at once. So proteins don't work in isolation, and now our maps show that. Let me give you an image that makes this concrete. HIV-1 protease, one of the most important drug targets in medicine, and it only works when two copies of the same protein lock together to form a functional enzyme. Now, before today, the AlphaFold database could show you a single copy, like having the blueprint for one jaw of a pair of pliers. You could study it, you could admire the engineering, but you couldn't really see how the tool actually grips. And Martin Steiniger, the computational biologist at Seoul National University who helped lead this work, described complex prediction as quite a different beast from the single protein work that came before. The computational intensity is extraordinary, and the collaboration required to pull it off, Seoul National University, EMBL, EBI, Google DeepMind, NVIDIA, this tells you something about the scale of the problem, that no single lab could have done this alone. Now, the reason this is important beyond structural biology is what it enables. When you can computationally predict how proteins pair and assemble, not just what an individual chain looks like in isolation, you can start to see drug targets that were structurally invisible before. Protein interfaces, the surface where two molecules actually touch, the binding pockets that only exist when the complex forms. These are the hinges of molecular life, and the prior database didn't include them because it couldn't. The Century Report has been tracking a cascade in this arc. Blood-based Alzheimer's prediction on February 23rd, Tanocyte clearance shuttles on March 9th, protein folding diagnostic signatures on March 12th, each one of these operating at a different scale through a different mechanism. The AlphaFold expansion doesn't add another finding to that list. It changes the substrate on which all those findings can even be developed. It's infrastructure, the equivalent of going from an individual roadmap to a satellite live view of the entire transportation network. And then the same day, two findings arrived that show what becomes possible when biological resolution crosses this kind of threshold. Researchers at VIB and KU Leuven identified for the first time exactly how lecanemab clears Alzheimer's amyloid plaques. Not approximately, not the general mechanism, but the precise molecular pathway. The antibody's Fc fragment, which is a specific structural region on the tail end of the molecule, reprograms microglia. These are the brain's own immune cells. It flips a switch. The microglia activate a specific gene program, including strong expression of a gene called SPP1, and they begin clearing the toxic deposits. Now, here's the critical detail. When the researchers removed the Fc fragment, the antibody still bound to the plaques. It still found its target, but nothing happened. No clearance, no microglial activation. The Fc fragment isn't helping, it's everything. Without it, the antibody is a label, not a treatment. And this opens a door that lecanemab itself can't walk through. If the mechanism is microglial reprogramming, if the antibody is essentially just a delivery vehicle for an activation signal, then the question shifts to, can you activate microglia directly without the antibody at all? This is a cheaper intervention and a much more targeted one as well. So potentially oral rather than intravenous, for example. The researchers are explicit that this is where the work points, not better antibodies, but direct microglial programming. And now the third piece. Harvard's Wyss Institute published results on a platform called DoraVac, a vaccine built from folded DNA nanostructures, DNA origami, tiny self-assembling shapes that present immune-stimulating molecules at precisely controlled distances down to the nanometer. The researchers tested it head-to-head against SARS-CoV-2 mRNA vaccines delivered through lipid nanoparticles, the same tech that powered the pandemic-era shots, and Doravac matched the immune response. That alone would be notable, but the advantages go beyond just equivalence. DNA origami is much more thermally stable than mRNA in lipid nanoparticles. The manufacturing is potentially simpler, and the platform is designed to target peptide regions conserved across multiple viruses. SARS-CoV-2, HIV, Ebola, which means a single platform architecture could address a range of pathogens without fundamental redesign. What connects these three? The AlphaFold expansion makes protein interactions computationally visible across the tree of life. The lecanemab finding makes a specific immune activation pathway mechanistically precise enough to redesign around. Doravac makes vaccine architecture programmable at the molecular level. Each one represents the same underlying compression that we're seeing right now in medicine. The distance between seeing a biological mechanism and an engineering intervention that just acts on it, that distance is shrinking, not gradually, but in discrete jumps. And each jump opens a design space that before we couldn't access. Okay, another story here. Let's move on to the electric grid. Different story, though, same structural logic. The physical infrastructure that sustains the intelligence era is colliding with the institutional architecture that governs it. The Energy Information Administration published an analysis today, projecting that data center demand could drive wholesale electricity prices in Texas up by 79% by 2027. That's the high demand scenario in ERCOT, the Texas grid. 79% in 2 years. Now, here's the structural detail that makes this more than just a scary number. The EIA modeled both ERCOT and PJM, the Eastern interconnection that serves the Mid-Atlantic that we've covered before, and parts of the Midwest as well. PJM sees price increases of roughly 4% under the same high demand scenario. ERCOT, 79%. The difference is not demand. Both regions are absorbing massive data center growth, but the difference is architecture. So PJM is interconnected with neighboring grids. So when demand spikes, it can import power. ERCOT operates on an island. It absorbs everything internally. So the same demand pressure that causes a 4% price ripple in a connected system becomes a 79% price shock in an isolated one. That's not an AI problem. That's an infrastructure design problem that AI demand is now revealing. ERCOT's isolation was a political choice made decades ago to avoid federal regulation, and that choice is now colliding with the demand trajectory that its architects never even could have imagined. PPL Electric's rate case settlement filed at the Pennsylvania Public Utility Commission shows what the institutional response to this collision looks like, but in practice. PPL's interconnection pipeline has swelled to approximately 20 gigawatts of contracted large loads against a current system peak of 7.8 gigawatts. That ratio, more than double the entire existing system demand, represents a growth trajectory that took a century to build, now being replicated in under 6 years. The settlement creates a new tariff category for data centers, 10-year minimum commitment, security deposits covering the full cost of grid upgrades they require, minimum load guarantees with exit fees if they leave, and $11 million a year into residential low-income energy assistance programs. This extends something Century Report has documented across multiple states. Illinois requiring data centers to supply their own renewables. Virginia mandating that utilities measure and optimize existing grid capacity. California ordering 6 gigawatts of new zero-emission generation. Each mechanism different. The direction, the same. The era in which massive loads could simply connect and let everyone else absorb the cost, that era is ending. And here's where I really want to push past where most of the energy coverage stops. The typical framing treats these tariffs and regulations as friction, obstacles slowing down the AI build-out, but that misses the trajectory when demand charges reach $70 per kilowatt, a number cited at a facilities management conference yesterday. They create powerful incentives for on-site battery storage, load management, and efficiency investments. Every data center tariff that requires interconnection cost absorption also creates a customer class with deep financial interest in grid modernization. The short-term regulatory complexity is producing the long-term architecture of a grid that can sustain not just data centers, but the entire electrification transition. The friction is the mechanism, not the obstacle. The mechanism. All right, let's shift to another story today. This is when generative systems meet actually vulnerable people. So we have three teenagers in Tennessee who filed a lawsuit against xAI alleging that its Grok image generation system was used to create sexually explicit material from their real photographs. The complaint describes a perpetrator who used a third-party application accessing Grok to morph the minor's social media photos, uploading the results to a file-sharing platform and traded them for explicit content of other minors. The victims' real names and school were attached to the files. One plaintiff even discovered that AI generated explicit images of herself and at least 18 other minors were circulating on Discord. I want to be really careful here because this story involves real harm to real children, and I want to be precise about what the legal claim is and what the structural question, therefore, is. The lawsuit alleges that xAI failed to test the safety of the features it developed, and that Grok is defective in design. xAI has maintained that anyone using Grok to generate illegal content faces the same consequences as uploading any illegal content directly. Now, those are two different frames, design defect versus user misuse, and the legal system is now being asked to decide which applies. And the answer will shape how every AI system with image generation capability is designed and how it's governed. On the same day, the Australian E-Safety Commissioner's correspondence with X, obtained through Freedom of Information laws, revealed that between January 1st and 15th of this year, X removed 4,500 pieces of Grok-generated content and suspended over 674 accounts for child exploitation policy violations. The regulator described child sexual exploitation material as particularly systemic on the platform and more readily accessible on X than on any other mainstream service. Now, set that next to Anthropic and OpenAI. Anthropic posted a job listing for a chemical weapons and explosive defense expert, specifically to prevent its systems from enabling catastrophic harm. OpenAI has posted a similar role at nearly double the salary. The distance between these two approaches, building domain-specific safety expertise before harm occurs versus facing litigation after it's already spread, is the distance the entire field must traverse. And what we're watching is that distance being measured in real time through lawsuits and hiring decisions and regulatory correspondence. On March 14th, the Censure Report documented a Lancet psychiatry review identifying clinical categories of AI-associated delusions and naming Claude as the only tested platform that refused to assist with violent planning. The xAI case adds a harm category that clinical taxonomies haven't even addressed yet when it comes to AI. No unified governance framework coordinates these responses, not yet. What exists instead is a patchwork: litigation establishing precedent, safety hiring building internal capacity, clinical research building taxonomies, regulatory bodies filling FOI requests. Each piece advances independently. Each one fills a part of a vacuum, and the framework that will eventually emerge is being written through exactly these kinds of cases, precedent by precedent, hire by hire, incident by incident. The question is, how many children's photographs enter trading networks before the framework is finally complete? Okay, here's what I would really like you to carry out the door today. A database of 200 million proteins just gained the ability to show you how those proteins actually work together as complexes, as machines. A research team identified the exact molecular switch that makes the most promising Alzheimer's drug function, and in doing so, showed that the drug might not be the point. The switch might be activatable directly. A vaccine platform built from folded DNA matched the immune response of the technology that helped us to get out of the pandemic, while being more stable, more programmable, and designed to work across multiple viruses simultaneously. That's 3 findings in 1 day. And in each case, the advance wasn't discovering something new. It was seeing for the first time something that was always there. The protein complexes were always forming. The microglial program was always waiting to be activated. The DNA origami was always foldable into vaccine architecture. What changed is the resolution: computational, experimental, molecular. The instruments finally matched the complexity they were pointed at. Very soon, watch for the downstream applications that flow from the AlphaFold complex database, because 1.7 million homodimer predictions across 20 species isn't a paper. It's a new starting condition for drug discovery, disease research, and synthetic biology. The tools built on top of that database in the next 6 months will tell you more about the pace of this transition than the database itself. The infrastructure is laid. Now, watch what gets built on it. We'll see you tomorrow.